But in eukaryotes, right, we're making these mRNAs and then we're sending them off to ribosomes other places. So we've got to have this little RNA make it there. So we add some stuff onto the ends to kind of protect the mRNA, right? We put this little 5' cap on here, on the 5' end, and then we put a whole bunch of adenine, right, a poly-A tail on the 3' end to kind of just like keep the ends from getting busted up while it's going to where it's got to get to, right? So that was one thing, right? Because we talked about, we were kind of getting to this point where, because of the way prokaryotes do their thing, right? Coupling transcription and translation, right? It's just all a very direct process. But in eukaryotes, it's less direct. We're going to cap it, and then we're still not done with the mRNA. It actually still doesn't code for a protein, right? So we've got kind of one more step of processing this mRNA, right? That's called splicing. So we've got to talk a bit about the relationship between like genes in your DNA and then the actual proteins we're trying to make. So in prokaryotes, genes are collinear with protein products, right? So we transcribe a piece of prokaryotic DNA into RNA, and then we slap a ribosome on that and it makes a protein, right? And you're done. Eukaryotic genes aren't like that. So I've got my gene in my DNA, but it's actually not all gene. There's like chunks of other stuff stuck in it, right? And so it's this mix of things that code for the protein we want with pieces of things that don't code for anything necessarily stuck in between pieces, right? So we call this pre-mRNA. So while it's still got these non-coding chunks stuck in it, right? The non-coding things are called introns. They're things that are just stuck in there, right, in the DNA. And then we have the things that actually code for the protein you want to make, and those are called exons, right? The pieces that are going to get expressed, right, actually made into a protein, right? So all of your DNA is a combination of coding DNA, right, that codes for a protein, these exons, with these bits that probably don't do anything, these introns. And so when we transcribe this into mRNA, right, we still have the same problem here. We've got our caps on the end, but we still have these pieces that don't code for anything and are just going to get in the way when we try to make, right, the protein using the information in the coding bits. So we're going to do what's called splicing, where we cut out the non-coding pieces and then put all the bits that actually code for a protein together, right? So primary transcript has to be cut and put back together to make mature mRNA, or mRNA that's actually ready to make a protein, right? And this is called pre-mRNA splicing because we're splicing the pre-mRNA, fine. And this is done by small nuclear ribonucleoprotein particles that help us, which are called SNRPs. No lie, some scientists decided these are SNRPs, right? And a SNRP can recognize the boundary between a piece of coding mRNA and non-coding mRNA. So, right, so where the intron meets the exon, right? They find that, they cluster there, and a cluster of SNRPs is called a spliceosome. This just sounds like I'm making up words, right? Which splices and removes the introns. Looks something like this. So we have our little pre-mRNA. I've got bits, right? Exon 1 and exon 2 that actually code for the protein I want, with this long string of junk in the middle, basically. So these little snRNAs clamp on at the boundaries, right, at the end of the non-coding stuff, and kind of pull it together. So they kind of cinch up all this stuff that we want to cut out, right? And all these guys together are the spliceosome, right? All these SNRPs and associated proteins together, right? Tie up this intron, right, this non-coding piece, and they kind of twist it off, right? So they're going to kind of loop it around. They call it that piece a lariat, right? So we've got our coding bits. We're going to kind of loop this up and pop it off, right, and send it away, and then take exon 1 and exon 2 and stitch them together. Now we have mature mRNA that only has the code for a protein in it. Now, one extra layer kind of interesting on this. So we can cut out these non-coding pieces. We have these coding pieces which we put together, right? We splice them back together. But in eukaryotes, you can put these, I mean, it makes sense, right? I pull out all the non-coding parts and I can put everything back together in exactly the order it was, or maybe I shuffle it around a bit. Maybe I don't use all of it, right? It's called alternative splicing, right? There's alternative ways. There's multiple ways to put these little coding bits back together, right? So a single primary transcript, right? A single pre-mRNA, once you cut the introns out, can be spliced into different mature mRNAs, depending on how you put the pieces back together, right? And if you put the pieces back together in different orders or with different kind of members present, you will get different proteins, right? You're just coding for different amino acids, you make a different protein, right? So that's called alternative splicing, which means we kind of add levels onto the amount of information in our nuclei, right? Because you have all your DNA in your nucleus, that's your genome, right? It's got all your genes in it. And so your transcriptome then are all the RNAs that can be transcribed from your genome, right? Because not all of your DNA gets transcribed. Right? There's some stuff that we just never use. And so we can make these transcripts. But because we can take each of these transcripts and maybe make a few different things from each of them, you kind of can make, basically you can make more proteins than you can make transcripts. So all the proteins that your genome is able to produce is your proteome. All right. So there's a couple, well, a few kind of, I don't know, results, conclusions, sorts of things that come because of the way we process our genes, subsequently transcripts, subsequently proteins, right? One thing is a lot of human genetic disorders come about this way. Because once you kind of add complexity to a system like this, right now I'm chopping this transcript apart and putting it back together. Maybe I put it back together different sorts of ways. It allows for some plasticity or ability to adapt and that sort of thing, but it also means you've kind of opened up a new area to make mistakes, right? If you put a protein back together wrong, right, then you've got a problem. So there are also genetic disorders that can be associated kind of with alternative splicing making a mistake, right? But it's also super common, right? It's estimated around 95% of mammalian genes undergo alternative splicing, right? Well, and it makes sense, right? Because your genome has about 20,000 genes in it, but you're able to make around almost 100,000 protein coding transcripts, right? All because of this ability of our cells to do this alternative splicing of pre-mRNA. All right, so we can make mRNA. What good does that do? Well, we've got to make it into a protein. So the next chunk of chapter 15 is about getting from mRNA to protein. We're going to need a couple other guys to help us out with this. So we're needing some tRNA and of course the ribosomes, right? Ribosomes from way back at the beginning of the semester are for making proteins. They are going to use the directions in the mRNA to make a protein with the help of tRNA. So what is tRNA? It's an RNA molecule. And it's an RNA molecule that specifically kind of, right here it is, kind of this little like busted four-leaf clover that twists up on itself into this little L shape. Right, so it's kind of got two ends. So this end is an anti-codon loop, right, with 3 nucleotides that will match the codons in the mRNA. The acceptor end basically has a little handle for holding on to amino acids, specifically the amino acid that matches the codon they're carrying, right? So we kind of draw them this way, this weird little, I don't know, weird looking guy. So he's got an acceptor stem for carrying an amino acid that matches the codon. He's carrying the anti-codon, right? Because he's got to have the complementary nucleotides, right, for the mRNA here. So tRNA is for transferring amino acids to the ribosome to make a protein. All right. We're going to have to add some more enzymes as we get in here. So amino acid tRNA synthetases add amino acids to tRNA at that acceptor stem, right? So we need an enzyme to put an amino acid on the tRNA, fine, right? So it puts an amino acid on the tRNA. It's right there in the name. That works well enough. And there are 20 of these synthetases, one for each amino acid, right? We've got 20 amino acids, right? And so each synthetase will be specific to amino acid that will attach to a tRNA, right? One for each amino acid. So every synthetase recognizes one amino acid, but frequently more than one tRNA, because why? What did we learn about the genetic code last week in terms of how it codes for amino acids? Starts with a D. Something about the genetic code that starts with a D. A D? No, it wasn't the dogma thing. So we were talking about kind of the nature of the genetic code and these codons, right? Every 3 nucleotides in your DNA codes for an amino acid. But the code is something. Yeah, I think so. Yeah, degenerate. The code is degenerate. Yeah. Okay. Right? Because, right, for example, right, we've got this amino acid arginine. There are 4 different codons, right, sets of 3 nucleotides in your DNA that all code for arginine. Right? So if you have a synthetase that recognizes arginine, it's going to match 4 different tRNAs, right? Because there's 4 different codons that could be for arginine. Right? And so it doesn't have to be picky about which one. It just has to get an arginine on there. Right now, the tRNA is going to take this amino acid to the ribosome and put it on the polypeptide. So it would behoove us to give this tRNA a little oomph so that it's able to do so. So we're going to have this charging reaction that's going to happen, right? Where we're going to add an amino acid to tRNA, spend an ATP so that when the tRNA gets to the ribosome, we don't have to put in any more energy. It'll just be able to make the little peptide bond, put its amino acid on the protein, and we're good. Right? Now, all of our kind of quality checking in this process is done by the synthetases and the tRNA. Ribosome does not verify amino acid on the tRNA. It assumes if the tRNA shows up with the right codon, it's got the right amino acid, right? And it just goes on with its business. So don't worry too much. What we're looking at here, this is the synthetase. Here's our amino acid, tryptophan in this case. We're going to spend an ATP to pop this tryptophan on here. Here comes our tRNA with the anticodon for tryptophan, right? It's going to pop onto the synthetase and we're going to attach the amino acid to the enzyme, right? Now we've got this kind of charged bond on this tRNA that codes for tryptophan. So it's going to head on over to the ribosome and add that amino acid where appropriate. So the ribosome then is step next. So the ribosomes are where translation happens, right? It's where we make proteins. We covered that ages and ages ago. And it's going to have the same 3 stages as basically everything else, right? DNA synthesis, transcription, and translation all have these same 3 stages: initiation, elongation, and termination, right? And in translation, this all happens in the ribosome, okay? So things we already knew about the ribosome, right? It's got a large and a small subunit that come together when we need to translate mRNA into protein. But it also has in these 2 subunits 3 binding sites for tRNA. We're going to spend some time talking about those guys. So it's got an A site, a P site, and an E site. A site is where the amino acid comes in. P site is where the polypeptide chain is. E site is where the tRNA is going to exit, right? So in order and naming, they're relatively straightforward, right? So the tRNA that we've just charged up with our charging reaction by the synthetase is carrying its amino acid. It lands in the A site. It forms a peptide bond with this guy in the second site, pulling the peptide chain, adding its amino acid to it. And then the guy that was in the P site is now empty, moves to the E site, and eventually gets booted out the back end of the ribosome because he's done. So let's look at that. All right, here's our ribosome. We've got mRNA to handle, so we've got a small subunit and a large subunit that are going to come together on this mRNA molecule, right? And it's just going to kind of be trucking down the mRNA matching, right, a tRNA with a specific codon to add its amino acid to the polypeptide chain. And then the ribosome is going to kind of shift down. These guys are going to shift one step to the left. We'll bring in another amino acid over and over again. So, all happening in the ribosome. Ribosome has two functions. Decode mRNA, right? Because mRNA is carrying the instructions for the protein. This is happening in the small subunit. And then up in the large subunit, we're going to actually be making the peptide bonds, right? We're going to be bringing the amino acids in and kind of clicking them together as we move down the mRNA, kind of reading the instructions for building this protein, right? We're going to need a transferase for this because we're going to be transferring these kind of peptide bonds from one tRNA to another. We'll see that. We've got a nice big diagram of the whole thing together in a second. But the peptidyl transferase is kind of a component of the large subunit. So it's all the amino acid polypeptide stuff is happening up in the large subunit where we're making these peptide bonds between each of these new amino acids as they come in. Okay, so we have all the pieces. So what does the actual process look like? You kind of mentioned it. So we'll walk through like we have walked through initiation, elongation, and then termination. So in initiation, right, we'll start with the prokaryotes because they're a little simpler. So we've got the initiation complex. So we've got initiator tRNA, right? So we've got one tRNA that's going to kind of get the whole thing started. He carries an N-formal methionine. We might remember from Friday that methionine, AUG, is the start codon. So it's going to be at the beginning of our mRNA. So it's a nice place to kind of line everything up in the ribosome, right? So this is going to kind of all be down in the small subunit of the ribosome, right? Because this is where the mRNA is going to come in. We want to make sure everything's lined up before we start building a protein, right? And in prokaryotes, we've got this ribosomal binding sequence. We've got our mRNA, and there's this little sequence that's like, hey, ribosome, latch on here, the start codon's coming up next, right? So it kind of lines the mRNA up inside the ribosome. Then we tack a large subunit on, right, which has all of the protein putting together pieces in it, right? And then that initiator TNA with its methionine pops into the P site, the peptidyl site, leaving the A site empty for the first tRNA to show up with its first amino acid, right? So like this, right? Small subunit, here comes my mRNA, right, coming away from the DNA. Got my initiator tRNA hanging out over here. So we start to kind of put everything together, right? The mRNA slides in, tRNA pops on to the start codon, like this is where we're going to start. Large subunit lands on top. So we've got our start codon in the P site. We're waiting for this first amino acid, whatever this here codes for, to come land in. So we've got everything put together to make a protein. All right. Eukaryotic initiation, similar, but different as always. So instead of having some weird amino formal methionine, whatever, we just have a regular old methionine. So regular tRNA for the start codon, AUG with a methionine amino acid attached is going to land in that P site. We're going to have a more complicated initiation complex. There's always just kind of more moving parts in a eukaryote. That's fine. And we don't have this ribosomal binding sequence because remember we have on our transcriptome, or transcript rather, this little cap, right? That little 5' cap. And it's going to work like an RBS. The small subunit's going to recognize the cap and pull that end, that 5' end, into the ribosome, right? So we can initiate. Elongation is just adding amino acids, right? So I've got my start codon in on that P site, the second one. tRNA with my next amino acid comes, lands in the amino acid site. And this little guy is going to take his amino acid. He's going to put it on top of the guy in the A site, and then everybody shifts to the left, right? Next guy comes in with amino acid, takes this whole little bit, pops it on top, attached to the amino acid he was carrying, everybody moves to the left. And that just happens over and over again, right? Right, so like this. So we've got our little start codon guy. He's carrying his little methionine. Our first amino acid, whatever this codes for, it's a secret, right? Comes in here. And he's just going to take our current peptide chain and just pop it on like a little hat. Now we've made this peptide bond, we've linked these two amino acids together. And so the next thing that's going to happen is the ribosome is going to move down to the next codon, and these guys are all going to shift left. Shift left. Empty guy is going to go into the empty site. Guy with the peptide chain is going to move into the P site, and we're ready for the next amino acid to come in, right? And that just happens over and over, right? So matching tRNA with the anticodon to the next mRNA codon, forming a peptide bond, shifting everything down, right? Just in a circle, right? So we've got this little helping elongation factor, brings this guy in to the A site. He's got his little amino acid. We've got this forming polypeptide chain, right here. I'm gonna pop him over here onto this guy. Everything's gonna shift left. So now here's our growing polypeptide chain. This guy's now empty, so he's gonna get booted out. And eventually we'll have our next tRNA that matches whatever our next codon is. And then we do the whole thing again, right? So it just kind of goes on and on and on until you got to the end of the mRNA. At which point you've created a nice long polypeptide chain that will fold up into a protein. Now, another note. So there are fewer tRNAs than codons due to this kind of thing that happens called wobble pairing. So if we remember that big table of amino acids, frequently for something like, say, arginine, right? I think arginine starts at cytosine uracil. You don't have to remember this. Cytosine uracil, but then that third position, right? The third nucleotide, it didn't matter what it was. It could be any of the 4 nucleotides and you would still get a uracil at the end or a arginine at the end, right? And so this means that we can have tRNAs, particularly tRNAs that code for amino acids that say have 4 different codons that code for them. And they can basically just be really not picky about that third nucleotide, right? Because it doesn't matter. It's all going to give you the same amino acid, right? And this is called wobble pairing. It's kind of like a less accurate pairing, right? Which means we don't have to have as many tRNAs as we have codons because there's like 100 and something of them, right? And so we can kind of have fewer tRNAs bopping around. It simplifies things at least a little bit. So how do we stop? Fortunately, right, we've also got stop codons, right? UAA, classic stop codon. And so there's not a regular old tRNA that matches the stop codon. There's actually a release factor. So the ribosome's like trucking down the mRNA, you get to this stop codon. And instead of having another one of these tRNAs with an amino acid, we've got this release factor that then comes in and matches to the stop codon, which stops translation. And we release the polypeptide chain to then go and fold and become a protein. And then all the pieces dissociate, right? Because we're done translating. And ribosomes, when they're not translating, are kind of floating around in parts. So, okay. There's another thing we know. We may not remember that we know it, but we have known it, right? Proteins can be made anywhere that there are ribosomes, right? And in our cells, ribosomes are kind of everywhere. They're in the cytoplasm, they're stuck on the sides of the rough ER, they're kind of all over. And so translation then can happen in any of those places. We can make proteins any of those places. So how does our cell know when a protein that's being made needs to go into the rough ER, right? Because remember, things go into the rough ER, into the endomembrane system, and they get processed into specific things that your cell's going to send off, right, in little vesicles to go do work. So proteins that need to go into the endomembrane system have a little signal sequence at the top, right? So you've got a ribosome, mRNA is going through, the polypeptide chain is kind of coming out of the large subunit as we put it together. And these first few amino acids, there's some of them that work as a little signal, right, as they come up out of the ribosome. And so if a peptide chain has one of these little signals up on the top of it, a signal recognition particle, aptly named, will come find it, right, kind of grab onto the peptide chain and plug it into the ribosome so that that protein then is getting made into the endoplasmic reticulum, right? Like this, right? Got my little ribosome. It's cranking out this polypeptide chain. which happens to have these first 3 amino acids that draw the attention of the signal recognition particle that comes in and grabs it, docks it into the rough ER so that now this protein is being made directly into the endoplasmic reticulum, right, where it can go through the endomembrane system and get processed accordingly. All right. Summary, right? Because all of this is just gene expression, right? This is getting genes from just being kind of information coded for in your DNA into actual proteins in your body that will do jobs, right? And that is all gene expression. So gene expression is that converting genotype to phenotype, right? Genotype determines phenotype. And we've just basically walked through the actual kind of nuts and bolts process that gets you there. So transcription, right, produces a copy of your DNA in mRNA form, which then directs synthesis of protein, right, by the process translation. Both transcription and translation, just like DNA synthesis, can be broken down into 3 stages: initiation, elongation, termination, and as one would expect, eukaryotic gene expression is more complex than what we see in prokaryotes. Right? So just kind of summarizing all of that. Now this is kind of for you. So it's just kind of got all of those pieces put together, right? Here's our transcription process we talked about. Here's the splicing, the putting together of the ribosome, and then the process of creating those proteins, right? So it's just kind of putting all of these figures and things we've been talking together, right? Because they're all part of one process. We've just kind of broken it apart, kind of take it piecemeal in. But it is a single kind of continuous process, right? Getting from your DNA to a finished protein. So that is kind of there for your studying purposes, as is this, right? Because as we've gone through, we've kind of like been mentioning all of these ways that gene expression is different in things like bacteria versus us, right? For example, introns, those non-coding pieces, prokaryotes don't have them. Most of our genes have them, right? So this is just kind of a summary slide of those sorts of things that we've already talked about if you want to kind of see it all in one place, right? So we've got this kind of brought together. All right. The next section, we'll do a little bit, is talking about mutation, right? So mutations are just altering of our genes, of our genetic code, either in the process of making proteins or kind of in the process of copying our own DNA, right, that gets passed on. So we're going to talk about a few different kinds of mutations and those and the effects of them. So a heritable change to genetic material is called a mutation, right? So something changes in your DNA. That change in DNA can be passed on to other generations. That is a mutation of your DNA. Even changing kind of one single nucleotide, right, can be debilitating, right? Can change an amino acid, can change the entire structure of a protein, right? Sickle cell anemia that we've been talking about, right? Kind of at the beginning of this chapter. All that sickle cell anemia is changing one adenine to a thymine in your DNA. It's just an A for T, single nucleotide change, which is enough to change the entire structure of your hemoglobin, right, and cause an entire syndrome, right? So when you change the A to a T, in this case, you switch out the amino acid glutamic acid for a valine, causes your beta chains to kind of aggregate, clumps up this protein, and changes the shape of your red blood cell, which is what causes, right, the actual kind of disease and symptoms of sickle cell anemia. Oh, I thought I had another slide in there. I guess I don't. All right. And so that alteration, that deletion of just a single base, right, one little nitrogenous base gets swapped out, is called a base substitution. Easy enough. You substitute one base for another. It's called a base substitution, right? And so these are all point mutations, right? Just kind of one little change, right? So we can change one base for another. These substitutions can be silent, right? Particularly if you mutate the third nucleotide, right? The one that can kind of be different and you still get the same amino acid. So if you mutate, right? Switch out one nucleotide for another, but you still get the same amino acid, you're gonna get the same protein. That mutation is silent, right? Because there's no kind of outward produced effect of changing that base. But if it does change the amino acid, we call that missense, right? The code doesn't make proper sense anymore. We've changed the thing that we're making. These can take the form of transitions or transversions, right? Remember, we've got T's and C's, which are pyrimidines. So if you switch a T for a C, that's a transition. If you switch an A for a G, right? Because you remember purines have a certain shape, pyrimidines have a certain shape. So you're kind of, it's less different is kind of a way to think about it. Now, if you switch out pyrimidines for purines, which is what sickle cell does, then you're really kind of changing the shape of things. And you end up usually with kind of more drastic kind of outcomes for transversions. Last, you can have a nonsense mutation. So if you change a base in a codon and it turns it into UAA, turns it into a stopcodon, right? Then you've just busted the gene completely, right? Now, like when you're making this into a protein, you're going to hit a stopcodon halfway through and the whole thing's going to just kind of fall apart, right? So you can have a silent mutation, which doesn't change anything. You can have missense, where you put in a different amino acid and change the shape of your protein. Or you can have a mutation that puts in a stopcodon and you've just broken your gene. This is just kind of, if it helps you to kind of see what's happening, right? So if we have a missense, we've changed our arginine to a, what is that, a histine, right? We can add a stopcodon and then nothing will get made kind of after that point. So we can have point mutations where a single nucleotide change, right? And there's sickle cell, which is where, this will be the last thing we do. Where glutamine, right, we switch an A to a T, we get a valine. We're supposed to be making a protein that looks like this. We end up making a protein that looks like this. They all get stuck together and you end up with a sickle cell red blood cell just from changing one little nitrogenous base out on this huge molecule. So it's kind of crazy how large an effect such a small change can have. But I think that's good for today. Yeah, we'll talk about other types of mutations on Wednesday.